Hi, I'm Shefali, and I'm a student from the Campus Lab of Computational Biology at UC Santa Cruz. Today, I'll be discussing our paper on identifying selections operating on HIV-1 reverse transcriptase via uniform manifold approximation and projection. The contributors to this work include myself, Professor Manel Camps, and Jay Camps. So moving into the slides, we'll first talk a little bit about the goal and the purpose behind this project. Reverse transcriptase, or RT, is the target of two major classes of HIV antiretroviral drugs, NRTIs, which compete with the TNTPs for incorporation, and NNRTIs, which bind at the hydrophobic pocket, interfering with DNA synthesis. Unfortunately, resistant mutations of HIV have evolved. The evolution of antiretroviral drug resistance is facilitated by HIV's high mutation rate. Individual diagnostic mutations have been identified in HIV-RT that are predictive of resistance to specific RT targeting antiretroviral drugs. However, the evolution of these mutations and their effect on resistance is likely dependent on the specific sequence context within each HIV-RT sequence. This is due to the non-additive or epistatic effects that mutations frequently have in the presence of other mutations, an effect that is particularly prominent when a gene is under selection to evolve a new biochemical activity, which in this case is antiretroviral resistance. Our understanding of this context is limited by the large number of possible combinations. Several efforts to map the epistasis of HIV's drug resistance mutations have been reported using linear dimensionality reduction methods such as principal component analysis and multidimensional scaling. Linear dimensionality reduction limits the identification of large, interdependent networks of mutations which may be evolving together. Here we turn to uniform manifold approximation and projection, or UMAP, which is a recent nonlinear dimensionality reduction technique. It works by applying an n-wave analysis based on the elasticity of the manifold in n-dimensional space at the points made up by the sequences. This nonlinear dimensionality reduction technique allows the identification of relationships between an unbounded number of mutations simultaneously. The 72,200 HIV-1 reverse transcriptase sequences used in this analysis were sourced from the Stanford HIV drug resistance database. These sequences were taken from patients involved in longitudinal studies and include different treatment regimens. For each sequence, we had the patient ID, study year, and a list of the drugs administered to the patient for the purpose of HIV treatment prior to sequencing. Here, we'll refer to this additional information as the sequence's metadata. Following some quality control filters, such as the removal of deactivated viruses and incomplete sequences, we were left with a total of 14,651 sequences. This data was then prepared for analysis by splitting into the training data and the metadata. We finally one-hot encoded the training data and the sequence data to prepare for analysis. UMAP was utilized to obtain a projection of the training dataset onto lower dimensional space. In this lower dimensional space, each sequence is represented by a single point. Density-based spatial clustering of applications with noise, or dbScan, was chosen as the clustering algorithm for this analysis, as it does not require specification of a fixed number of points or sequences per cluster. In our analysis, this configuration resulted in 21 distinct clusters, 2 of which were considerably larger than the others. For each cluster, we now had a group of sequences which had been brought together based on some latent factors identified by the algorithms. We performed several tests to assess the biological relevance of the clusters that were obtained. The first one was to measure the phylogenetic relatedness of the clustered sequences. To this end, we computed a UPGMA tree for each cluster. The maximum depth of the tree represented the distance of the furthest removed descendant of the common ancestor, and thus this was used as a metric for comparison between the trees. The results are shown in this table below. As expected, we observed that the sequences in our clusters tended to be more phylogenetically related than randomly selected sequences, up to 14-fold. This is significant because phylogeny was not directly used to define our clusters. Another way to look at the ability of our analysis to identify related sequences is to look at the penetrance of all of the mutations found in each cluster. To measure the consistency in the representation of mutations within a given cluster, we define a homogeneity index as lambda 50 over lambda, where lambda 50 represents the number of mutations with the frequency greater than 50%, and lambda represents the total number of mutations observed. The result is shown in this column, and as expected, we see a substantial negative correlation between the maximal UPGMA tree length and the homogeneity index. In other words, this means that the more related the sequences are, the more likely a given mutation is to be highly represented in the cluster. We hypothesized that some of the clusters could identify distinct selections or distinct adaptive solutions to selections. Here we show the correspondence between mutation and treatment profiles. Each row represents a UMAP-derived cluster of HIV-1 RT sequences. Shown are the frequency of mutations by position on the left and the frequency of prescribed drugs in the treatment regimen for each patient on the right. To quantify the consistency in the treatment, we defined a treatment homogeneity index similar to the mutation homogeneity index. Here we show clusters with a treatment homogeneity index above a random control. Interestingly, we observed that these 8 clusters of sequences identified patients by consistent drug treatment. Here we plotted the correlation between the mutation homogeneity and treatment homogeneity indexes. Strikingly, they correlated strongly with an R of 0.83. This suggests that some of our clusters are identifying specific treatment regimens. However, when we looked at the profiles corresponding to each of these clusters, known diagnostic resistance mutations were largely absent. In an effort to show the effect of drug selection, we looked at cluster 2 sequences because we had a good representation of patients that were sampled before and after treatment. The results of this analysis are shown in Figure 5. In this analysis, we see a high representation of the main NNRTI resistance mutation, K103X, and a lower representation of 2 additional NNRTI mutations, Y181X, G190X. We also see NRTI mutations represented, notably M184X, D67X, and K70X. These results suggest that the UMAP-derived signatures that we identified are largely reflective of the network of interactions with the adaptive mutations, rather than by adaptive mutations themselves. By the end, we've used UMAP analysis to embed HIV-RT mutation data in lower dimensional space. We've visualized these results using an unsupervised density-based clustering analysis, which produced 21 clusters. 3 lines of evidence support the biological relevance of these clusters: phylogeny, chronology, and treatment. And remember to note that treatment information was not used as input in this analysis. The mutation profiles associated with each cluster likely represent the higher order epistatic context supporting these evolutionary pathways, which is information that's not generally accessible by other types of mutational codependence analyses. In the future, we plan to look at a few things. The generality of these observations needs to be confirmed in other models of adaptive evolution. Also, despite their association with specific drug treatments, our UMAP profiles contain almost no major drug resistance mutations. However, we still detect the emergence of diagnostic mutations in individual patients under treatment, showing that these viruses were under strong drug selective pressure. The apparent instability of these diagnostic mutations needs to be investigated further. This will be greatly facilitated by the availability of NGS sequence databases with deep coverage for individual patients. Also, our interpretation of UMAP profiles, or that they represent a network of mutually dependent mutations, needs to be confirmed, and the dependent structure and directionality of that needs to be dissected. This can be done through a combination of directed evolution and targeted mutagenesis approaches. Thank you. Here are some references. And yeah, any questions? Feel free.